video for our cellular respiration discussion. O remember, done na tayo sa Crash Course 5 discussion. So for this video, we are going to cover our Crash Course number 6. So recall that this is the 4th video for our Biochem Crash Course discussion. So from our CC5, we were able to cover first the principles of metabolic pathways. That's your bioenergetics, biological membranes, and membrane transport. Of course, secondly, we also covered the first process in your cellular respiration, which is your glycolysis. O diba? Simply the conversion of glucose to pyruvate via 10 individual reactions. And lastly, we also covered fates of pyruvate. And for our Crash Course 6, we will start our discussion with the TCA or the tricarboxylic acid, or also known as the citric acid cycle, followed by your electron transport chain, and lastly, ayan. So class, we also need to discuss the ATP accounting, so the net yield of energy from our cellular respiration process. So class, again, I hope similar from our previous videos that you are eager to learn and to add essential information to our biochem discussions. All right, so let's first discuss citric acid cycle. I think this meme is very famous, di ba? Very forgettable kasi si Krebs cycle. So I think from my personal experience, citric acid cycle is hard to memorize or familiarize because hindi straightforward yung reactions niya, aside from the fact na it is a cycle. And of course, marami rin kasing energy products and other coenzymes that participate in this specific cycle. But last, again, similar with glycolysis, meron tayong mnemonics. And very important lang naman here is that you know the regulated enzymes, the substrate, as well as the energy products. And with that being said, this is the overview of our citric acid cycle. Sir, is citric acid cycle catabolic or anabolic? Actually, guys, both. But for our first part of discussion, we will focus on the catabolic part of your citric acid cycle. So meaning, energy yielding pathway po ang citric acid cycle discussion natin firstly. So again, amphibolix ang process. So later on, we will discuss its anabolic role. But of course, oxidative catabolism ang process here in your citric acid cycle using your fuel molecules, si carbs, fatty acids, and amino acids. Sir, how about the energy products? Similar lang po ba siya sa glycolysis? Well, you can say that it's somehow similar, but here we add another electron carrier. O class, recall, here in our citric acid cycle, instead of ATP, si GTP po ang energy carrier natin. Okay? And aside from NADH, similar with your glycolytic pathway, we now have FADH2 as one of our electron carriers. O later on, you will see bakit naging preferred ng one of the reactions here sa citric acid cycle, si FADH2. O sir, how about the location? Of course, we have seen in our introductory or overview ng cellular respiration, sa mitochondria, nag-occur si citric acid cycle. And class, remember, strictly aerobic po si citric acid cycle because the enzymes need oxygen to function or to be activated here. Okay, so lastly, this is the overall reaction. Sir, so ang initial reactant natin ay acetyl-CoA. Yes po, along with other coenzymes in the form of NAD+, FAD, and GDP, and your cofactor, inorganic phosphate. Okay, so class, I hope you're ready. Again, we will discuss 8 reactions here. O sir, mas makonti pala siya compared to glycolysis. Yes, but again, it's not straightforward. Okay, so we will discuss in detail each reactions along with the intermediates that form on those reactions. Okay, so again, si TCA cycle po is a metabolic pathway that connects your carbs, fats, and protein metabolism. That's why we call it the central hub of our fuel molecules. All right, so now let's proceed with the specific reactions. Class, similar format lang tayo ng discussion with our TCA cycle reaction. So first one, of course, condensation. Diba, we know that it is a cycle, but for our discussion, we will linearize it. Recall, ang last product ng ating citric acid cycle is si oxaloacetate. So si oxaloacetate, abbreviated as OAA, is actually called as the initiator molecule here in our TCA cycle. So it is condensed with your acetyl-CoA via your citrate synthase reaction to form citrate. So here class, very straightforward ang reaction. Diba kasi first reaction pa lang, ang ating takeaway here is that the methyl group, ayan. So the methyl group of acetyl-CoA is converted to methylene in your citrate. Let's see the specifics. Again, ang process, aldol condensation. Di ba nag-appear na naman si aldol condensation here, which is followed by hydrolysis. Oh, so sir, mag-input tayo ng water. Yes po. So as you can see here again, key enzyme, citrate synthase. And class, again, here is our topic on your regulation. Si delta G ng first reaction, large and negative. Oh sir, alam ko na, regulated si citrate synthase. Yes, and this is actually the first irreversible step of our citric acid cycle reaction. Oh class, please don't forget ang regulatory point, we discussed it last Crash Course 5, is your pyruvate dehydrogenase complex. So yung mga reactions here are just regulated. Alright, so number 2 reaction, ito class is a coupling reaction, dehydration and hydration via formation of your intermediate. Class, ito ang specifics ng reaction natin. The process is isomerization through dehydration and then subsequent hydration to form isocitrate. O, diba? From citrate, we formed isocitrate through your cis-aconitate intermediate. Actually, much easier to familiarize ang enzyme here kasi similar lang siya ng name with our intermediate. So here, ang key enzyme natin is aconitase. So, sir, what happened here? What happened, Bella, diba? So here, you can see that citrate is converted to isocitrate via the interchange of your hydride and hydroxyl molecule. O, diba? Very easy to understand. Ang function here is to activate the decarboxylation reaction. O, sir, nasan yung decarboxylation reaction dyan? O, class 1 atat. That's our third reaction of the TCA cycle. Okay, so ready na with our first oxidative decarboxylation reaction. So this is the third reaction. Check the mechanism. Hindi siya similar sa pyruvate dehydrogenase reaction. Okay? So here we have a lot of intermediate. Ang daming nangyayari simultaneously. Alright, but class here again, I would like to emphasize, you just need to remember si isocitrate and si alpha-ketoglutarate. But for the sake of our discussion, of course, for you to better understand what happened here, ayan. So we will discuss the step-by-step reaction here on our third reaction ng TCA cycle. So here are the information. Process again, oxidative or oxidative decarboxylation. And of course, as you can see, very obvious naman, we liberate carbon dioxide. That's why ito class I, shortening ng carbon chain. So you can see, si isocitrate has a 6 carbon, while ang ating final product, alpha-ketoglutarate, has only 5 carbons. Because again, na-liberate natin yung 1 carbon through production of your carbon dioxide. Okay, so thirdly, of course, important class, na you remember here that we also produced NAD+. NADH in the process. So let's discuss the details of this third reaction. First one is actually an oxidation reaction. Remember class, sa PDH reaction natin, simultaneous ang oxidation and decarboxylation. Here in our oxidative decarboxylation ng isocitrate, it is a stepwise reaction. First, let's oxidize first your isocitrate. So remove natin yung hydride molecule via transfer to our electron carrier. So class, here I would like to emphasize, we can use either NAD plus or NADP plus. So yung NADP plus, may additional phosphate lang siya sa structure niya. Okay? So here, it depends kasi sa isozyme ng isocitrate dehydrogenase. So again, class, if you are not familiar with the full name ng ating mga coenzyme, again, please visit your manuals. At the end of our biochem notes, we have a list of abbreviations there. Going back, here, after oxidation, we produced an intermediate named oxalosuccinate. O sir, parang may co-factor here. Yes, okay? Si Mn2 plus will help stabilize your carbonyl group kasi ang next reaction natin, decarboxylation na po. Okay? So that will help the liberation of carbon dioxide here. So decarboxylation is again facilitated by electron withdrawal with the help of our Mn2 plus. So here class, yung dashed or yung broken lines here, meaning coordinated po si Mn2 plus to two different oxygens. So I hope that's clear. So ang third specific reactions natin for our isocitrate dehydrogenase reaction is just rearrangement. So mag-rearrange lang yung enol intermediate natin to form alpha keto glutarate as our final substrate of the third reaction. Class, check ang delta G value, negative 21. Ooh, so sir, related enzyme si isocitrate dehydrogenase. Yes po. Okay, so remember, this isocitrate dehydrogenase reaction is the first oxidative decarboxylation reaction. Okay, so class, kaya pa? So if it's too much or saturating na, so you can take mini breaks. So sa aking on-site classes, I give them 1 minute break interval for them to better absorb our discussion. Okay, so now we will proceed with our fourth reaction, another round of oxidative decarboxylation. O class, medyo nangangalahati na tayo sa reactions ng citric acid cycle. Here, your fourth reaction is the second oxidative decarboxylation. O sir, pa-review, ano nga po yung first oxidative decarboxylation reaction? Yung previous reaction, si isocitrate dehydrogenase. Class, si alpha keto glutarate dehydrogenase complex, similar po ang mechanism niya with our PDH. So sir, meaning simultaneous po ang oxidation and decarboxylation? Yes po. So that's why hindi na natin ito i-discuss in detail. Again, similar lang siya ng mga required coenzyme. We have TPP, we have lipoamide, and of course, we have FAD. So meaning, yung complex ni alpha Kg also has three components. Okay, for the process again, oxidative decarboxylation, removal of your carbon through liberation of carbon dioxide, and of course, meron tayong na-produce na another electron carrier in the form of NADH. So for class, we produced 2 NADH here in our TCA cycle. And of course, I would like to emphasize na here, we also produce thioester. So here kasi, ang coenzyme natin na OH, it is condensed to your succinyl-CoA structure. And ito class ay very important for our next reaction. Now, let's see later ha, I hope that you are excited. For our third irreversible step, again, ito po ay regulated as well. Actually class, in your TCA cycle, similar with glycolysis, we have 3 regulated enzymes. Oh, so sir, complete na natin. Yes. So let's recall again. First one, citrate synthase. Second, isocitrate dehydrogenase. And third, alpha keto glutarate dehydrogenase. Okay? So class, I would also like to point out na dapat at this point, you are all well familiar na kapag meron tayong dehydrogenase enzyme, they are responsible for redox reaction. Because again, the hydrogenases are types of your oxidoreductases. Diba? EC1 yun, or enzyme classification 1. Okay, so now let's proceed to the next half of our citric acid cycle reactions with our SLP. So this is the only SLP or substrate level phosphorylation reaction in our citric acid cycle. So you can see here, from our previously formed succinyl-CoA, we will need a coenzyme GDP in order to form the energy currency GTP, and of course, ayan, na-regenerate lang natin si OH from our previous reaction. O sir, ano po ang final substrate or final product? Succinate po. Okay, so for the specific process, this is a phosphorylation reaction where the energy that is conserved in your thioester where the energy of your thioester conserved in phosphoanhydride bond of GTP or ATP is formed. So as you can see here, class, again, very high in energy po kasi ang thioester. O sir, asan nga po ulit si thioester? Here, class. So it's like an ester functional group, but instead of oxygen, ang point of connection ni carbon is sulfur atom. Okay? And ang key enzyme, succinyl-CoA synthetase. Alright, very easy ang fifth reaction natin. For our sixth reaction, ito class, ang ating formation ng FADH2 electron carrier. Let's discuss. Your sixth reaction is a dehydrogenation reaction. These are the process and the key enzymes for this reaction. From succinate, we will use succinate dehydrogenase. Oh, so sir, redox reaction po siya? Yes po. So we will oxidize succinate to form fumarate, and simultaneously, we will reduce FAD to form FADH2, which is an electron carrier, through the transfer of 2 hydride groups from your succinate. Class, let's summarize the process. As you can see, here in our sixth reaction, we formed a double bond in the structure of fumarate. So this step is very crucial in order to regenerate oxaloacetate, our initiator molecule in the citric acid cycle. Okay? And of course, additionally, again, we produce FADH2. Class, this is very unique for our sixth reaction because si succinate dehydrogenase participates both in your TCA and your electron transport chain. Again, complex 2 po si succinate dehydrogenase in your electron transport chain. So you will see this later on our discussion on ETC. So this is the only TCA cycle enzyme present on the inner mitochondrial membrane rather than in the matrix, because lahat kasi ng enzymes of TCA cycle is or are located in the mitochondrial matrix. Si succinate dehydrogenase embedded siya sa inner mitochondrial membrane as our complex 2 in the electron transport chain. Class, clear ba with our sixth reaction? O sir, baka nakakalimutan mo, ano po yung reason why FADH2 is preferred here in this reaction? Because class, again, si FADH2 is low in energy in terms of its capability to transfer electron. So, most importantly, one electron lang po kasi ang kayang i-transfer ni FADH2. And that's why, hindi enough kasi ang energy conserved here in the structure of your succinate. That's why, it's strategic or it's more wise for our cells or for our enzyme to use FADH2 as our electron carrier. Okay? So again, class, ang formation ng double bond here will be very important in initiating methylene oxidation sequence on our next reactions. Okay, so now we arrive with our second to the last reaction. O class ha, konti na lang. For our seventh reaction, hydration reaction. So this reaction is a preparation step for our last step, which is regeneration of oxaloacetate. So here, initial substrate, fumarate, and we produce a transition molecule, carb and ion. O, diba? Negative ang net charge ng ating transition molecule. So here, we produce malate as our final substrate. O sir, ano po ang key enzyme here? Fumarase po. For the process, again, we just add stepwise your water molecule to introduce hydroxyl group in the structure. So this is the hydroxyl group in the structure of your malate. Again, its function is just to prepare our next reaction or the next oxidation step. Okay, now, ayan class, I hope that you are excited. We are now on our last reaction of the TCA cycle. Or class, straightforward, we will just convert malate to oxaloacetate with the simultaneous production of NADH. So far in total, we produce 3 NADH, diba? So 1 in isocitrate dehydrogenase, 1 in alpha ketoglutarate dehydrogenase, and lastly here in our malate dehydrogenase. Ang process natin here is oxidation. So here, oxidation of our hydroxyl group from our malate is important in the generation of our oxaloacetate. Here, if you're familiar with Claisen condensation, that is the specific reaction happening here. So I think you will discuss your Claisen condensation in organic chemistry. Okay, again, ang key enzyme, malate dehydrogenase. Dehydrogenase participates in redox reaction. Now class, this step regenerates OAA that will initiate another round of TCA molecule. Again, our last reaction is the most crucial here in order for our TCA cycle to be continuous, okay, for its continuity. Now, we will summarize our citric acid cycle. Class, again, I will flash the overall reaction. Here, important that you know the net energy produced. First one, 1 GTP, 3 NADH, and 1 FADH2 per pyruvate molecule, diba? Because again, si pyruvate is converted to acetyl-CoA. So sir, that means if we need to find, if we need to compute for the glucose, diba? For the glucose molecule, ita times 2 namin yung energy produced. Yes po. Because again, ang stoichiometry is 1 glucose molecule to 2 pyruvate molecule to 2 acetyl-CoA. That's why you will need to multiply the energy products by 2. Because again, here class is 4. So these products or energy products are for 1 acetyl-CoA molecule. Okay, so for irreversible steps, again, we have 3. Citrate synthase, isocitrate DH, and alpha ketoglutarate DH. O sir, pengin namang mnemonics. Of course, so here is our mnemonics, starting with the rated PG, o diba? Ayan, citrate is Krebs starting substrate for making oxaloacetate. O sir, baka meron kang alternative mnemonics. Of course, ready ako dyan, class. So familiar ba kayo? Kilala nyo si Tony Fowler? O ito, class. Can I keep selling sex for Mommy Oni? Ayan, so this is our mnemonics that is, well, relatively easier to memorize. So again, class, the initials of our mnemonics are from our TCA cycle substrates. Can I keep selling sex for Mommy Oni? Okay, so I hope this will be very helpful in your review session, particularly in memorizing or familiarizing your TCA cycle substrates. Okay, for the latter part of our discussion, let's first discuss regulation and then your anabolic role, and lastly, the deficiencies of your citric acid cycle. Ayan, so class, again, this is the visualization of our regulation. So don't worry, tinabilit namin yan because here naman hindi lahat ay important. Okay? So again, we have 3 regulated enzymes in citric acid cycle. So citrate synthase, the first reaction, isocitrate DH, the third reaction, and lastly, si alpha Kg or alpha ketoglutarate dehydrogenase, which is our fourth reaction. And to summarize the regulation, ayan, so we have here the inhibitors and activators of the 3 enzymes along with our committed enzyme of the citric acid cycle, si pyruvate DH complex. Oh sir, magte-take effect pa rin po ba here yung tip or yung trend na shinare nyo on our glycolytic pathway regulation? Yes po. Again, for our inhibitors, they are energy-rich molecules. For activator, energy-deficient molecules. Okay, let's discuss it in detail. Si ATP, NADH, and acetyl-CoA, they are energy-rich molecule. And class, in addition, acetyl-CoA is the product of your pyruvate DH complex. Oh, so sir, feedback inhibition din siya? Yes po. And lastly, we also have fatty acids. Because class, we will discuss in lipid metabolism, si fatty acids can be converted to acetyl-CoA. Okay? For activator, ayan, ang ating mga energy-deficient molecule, si AMP and NAD+. Alright, so si CoAsh, this is the coenzyme, di ba, of your pyruvate DH complex. So when we have enough concentration of CoAsh, of course, it will activate your pyruvate DH complex. And lastly, we have your calcium 2 plus experimentally determined po. Okay, so this is a cofactor. So very important yan in signal transduction that will activate our pyruvate dehydrogenase complex. So next, citrate synthase, or the first two, energy-rich molecule. Okay, the succinyl-CoA, this is a substrate of our TCA cycle. Remember class, the succinyl-CoA is a substrate that will form our GTP. That's why energy-rich molecule din po siya. And lastly, si citrate user, feedback inhibition, no? Yes po, because again, recall, citrate synthase product IC, citrate. Okay, si activator naman natin, energy deficient, of course, ATP. And for our next two enzymes, si isocitrate DH and alpha ketoglutarate DH, class, similar trend lang yan, ATP and NADH, energy rich, so they are inhibitors. For activators, ni isocitrate DH, ADP and NAD+. And lastly, si alpha KGDH, ayan, all of them are energy rich molecule. And lastly, activator, AMP. Diba? If you know the trend, if you are well familiar with the trend, madaling ma-familiarize ang regulation discussion. So class, let's briefly discuss the anabolic role of citric acid cycle. Because again, we said earlier, amphibolic po si citric acid cycle. And in terms of its anabolic role, class yung intermediates kasi ng citric acid cycle, so particularly your citrate, your isocitrate, alpha-ketoglutarate, and etc., they serve as precursors for biosynthesis of different biomolecules, particularly amino acid, di ba? So you will see this more on amino acid biosynthesis. Secondly, porphyrins, di ba? And lastly, fatty acids and sterols. So again, intermediates, they can serve as precursors for biosynthesis. And this sir, magde-deplete yung concentration ng intermediates once they serve as precursors. Yes po. Is that a bad thing? No, because we have anaplerotic reactions. So class, anaplerotic reactions are formally defined as reactions that replenish your TCA cycle intermediates. That's why very important that you are familiar with the term anaplerotic reaction. So I do hope that you were already that you were able to cover this topic on your undergrad biochem. So here, we tabulated the 4 known anaplerotic reaction. So here, I will give you a tip para hindi nyo na-memorize ang specific reactions. Ang tip ko here is just examine the reactant and the products of the reaction. For anaplerotic reactions, dapat wala po kayong makikitang TCA cycle intermediate. Okay? So ang TCA cycle intermediate must be in the product side. Let's check its reaction. Here in our first reaction, pyruvate, no, that's not a TCA cycle intermediate. Si PEP, hindi rin po. And si pyruvate, again, hindi rin po. So these two, pyruvate and PEP, are actually glycolytic intermediates. So clearly, pwede siyang anaplerotic reaction. So let's double check if these are really anaplerotic. So let's check the product side. So for our first reaction, it has an oxaloacetate. Okay? And class, remember, si oxaloacetate is an initiator molecule ng TCA cycle natin. Alright, so for the first 3 reactions, lahat yan, oxaloacetate. And for our fourth reaction, malate. Okay, class, so that's very easy to remember. I emphasize this because it appears in the board exam, particularly yung reaction ni pyruvate carboxylase. So ang question done is, which of the following is an anaplerotic reaction of your TCA cycle? Just use my tip. Okay, check the reactant, dapat lang TCA cycle intermediate. And then check the product as well, dapat nandun ang TCA cycle intermediate. Again, anaplerotic reactions are very important to replenish your TCA cycle intermediates because your intermediates serve anabolic role by means of being a precursor for biomolecule synthesis. Okay, and now we arrive at our last slide sa citric acid cycle. O class, di ba ang haba ng discussion on TCA cycle? So again, class, please find some time, your free time, in order to revisit our discussion. So for my tip here in Biochem, very helpful if you will use flashcards in your review sessions. Okay, sir, ibig sabihin sa deficiencies, meron din po pala talaga sa TCA cycle? Yes, but of course, they are extremely rare because again, very important kasi ang citric acid cycle enzymes natin as it is the central main energy harnessing pathway in our cell. So again, TCA cycle enzyme deficiencies are extremely rare. Alright class, so here are the deficiencies. So it's either the coenzyme or the anaplerotic reaction enzyme. Firstly, si thiamine, this is your vitamin B1. So thiamine or TPP is a coenzyme of your pyruvate BH and alpha-ketoglutarate BH complexes. O diba, abnormalities. Ang important here class is very. So this is a nutritional deficiency in TPP. And secondly, we have pyruvate carboxylase. O, diba? We discussed this on our previous slide, anaplerotic reaction enzyme for the regeneration of OAA. And of course, this will lead to lactic acidosis. O sir, bakit? O recall class, kapag kulang ang concentration or we are lacking pyruvate carboxylase, magde-deplete ang concentration ni oxaloacetate. Kapag nag-deplete ang oxaloacetate concentration, of course, mag-accumulate si acetyl-CoA. Remember our first reaction ng citric acid cycle, acetyl-CoA will be condensed with OAA. And thus, kapag wala na yung OAA, accumulation of acetyl-CoA will occur. And kapag nag-accumulate si acetyl-CoA, mag-i-inhibit ang pyruvate BH reaction. And of course, because of that, if a funnel ng ating cell si pyruvate into your lactic acid fermentation. And that will result to lactic acidosis. Diba class, you can see the interrelationship again of these pathways. So for hyperammonemia, we will discuss this on amino acid metabolism. Hypoglycemia, similar reason with lactic acidosis. And hyperalaninemia is due to glucose-alanine cycle. So for pyruvate carboxylase, it's important that you know the main abnormality, lactic acidosis. Okay, so class, we arrive at our quick quest to summarize our citric acid cycle discussion. Now class, let's see if you really absorbed our discussion on TCA cycle. First concept or first enzyme, isocitrate synthase. Secondly, isocitrate dehydrogenase. Third, aconitase. And fourth, succinate dehydrogenase. O class, these enzymes are those that participate in TCA cycle except one. Very obvious, I guess, di ba? We did not discuss ayan, si isocitrate synthase. So class, again, remember the enzymes and the substrates of your TCA cycle. Sir, how about the structure? Do we need to memorize it? No po. Only the name of the substrates and the enzymes. Okay, so last quick quest natin, glucose, citrate, succinate, and OAA. Naku sir, very obvious naman ang dali. Lahat ay substrate ng TCA cycle except glucose. Very good class. So now, we will proceed with our next topic. So you can pause the video if you need a breather, but we will move forward with our linking discussion to our electron transport chain. Class, as I have said a while ago, very busy po si mitochondria because again, 90% of the metabolic pathways occur here in your mitochondria. That's why it's very important that you are familiar with the structure of mitochondria. Alright class, here is the structure of our mitochondria. So very unique din kasi si mitochondria because it has several components. Let's dissect those components starting with outer mitochondrial membrane. So as you can see class, here is the outer mitochondrial membrane. So this contains special channels such as your porins or aquaporins for instance for water, making it permeable to most ions and small molecules. So somehow class, if we are going to compare your outer mitochondrial membrane with your inner mitochondrial membrane, less selective or relatively less selective po si outer mitochondrial membrane as it is permeable to most ions and small molecules. Okay, so next component, ayan, si intermembrane space. space. So class, I hope that you can see the structure. So this is the intermembrane space. So that's a fluid which is a space between your outer and inner mitochondrial membranes. So later on you will see that this is where the proton gradient is generated in our electron transport chain. Thirdly, ayan. So we are going to the innermost half. So inner mitochondrial membrane is our third component. So it has specialized structure, and of course, majority po ng ating enzymes are also in your inner mitochondrial membrane. That's why it's rich in proteins. And class, again, this is very selective because it needs to protect the inside of your mitochondria. So impermeable siya to most small ions and molecules. That's why meron lang tayo here specific transport proteins. So class, yung specialized structure of your inner mitochondrial membrane is your cristae. So cristae is your convolution. O diba? You can see that there are many folds in your inner mitochondrial membrane, and this is very important. Firstly, to greatly increase the surface area of the membrane, and secondly, of course, to compartmentalize different metabolic processes. Because again, 90% po kasi ng metabolic processes or reactions occur in your mitochondria. O you can see class, diba? Jam-packed si mitochondria ng different enzymes, complexes, and carriers. So class, important to know, your mitochondria also has ribosomes and porin channels. And lastly, ayan, ang innermost part ni mitochondria is your matrix. So this is a gel-like solution in the interior that contains different enzymes such as your TCA cycle, your beta oxidation enzymes, and lastly, si mitochondrial DNA. O diba? Again, si mitochondria has its own genetic information in your DNA. Ayan, for our next segment, we have a did you know portion of class. Did you know that outer space changes the mitochondria? So studies from NASA's twin study revealed that space travel actually alters mitochondrial gene expression. So when astronauts spend extended time in microgravity, it doesn't just affect their bones and muscles, but of course, in a molecular level, diba? It reprograms their cellular metabolism down to the mitochondrial level. In particular, ayan, upon microgravity on your mitochondria, it will decrease ATP production, it will disrupt oxidative phosphorylation, and of course, it can increase oxidative stress. All right, that's why for some reasons from your outer space, our astronauts have weakened immunity, increased fatigue, or there have been impaired tissue repair. All right, so again, ang bottom line, space doesn't just stretch time, so it actually also warps how our cells make energy. Diba? So class, please, kung pupunta kayo ng outer space, know the consequences. Of course, now, we will proceed with our linking discussion for your TCA cycle and electron transport chain through your mitochondrial shuttles. Class, recall, from our glycolytic pathway, we were able to generate 2 moles of NADH. And of course, saan ang site ng electron transport chain? It's inside your mitochondria. So kailangan punta or your NADH must be transported to your mitochondria. That's why, ang cytosolic NADH, they use shuttle systems that transport NADH electrons across inner mitochondrial membrane. And for this, we have 2 different shuttles. First one, we have the malate aspartate shuttle. So it is localized in your heart, liver, and kidney. So here, ang conversion natin is around 2.5 ATP per cytosolic NADH. Class, you need to remember this conversion. All right, and the second one, si G3P shuttle or your glycerol 3-phosphate shuttle, which is localized first sa brain and second sa skeletal muscle. Conversion, 1.5 ATP is produced per cytosolic NADH. Class, I would like to clarify, wala pong preference ang cells natin for these 2 shuttles because again, they are localized. For instance, sa heart, si malate aspartate shuttle lang ang available. For brain, we only have glycerol 3-phosphate shuttle. Okay, so again, that's for your cytosolic NADH. Sir, how about yung mga na-generate na NADH in your TCA cycle? Class, they are mitochondrial na. Okay, so they are readily oxidized for ATP production. That's why ang conversion natin is 2.5 ATP per mitochondrial NADH. So sir, what's the reason? Bakit when we use glycerol 3-phosphate shuttle, 1.5 na lang ang ATP compared to your original 2.5 kay malate aspartate shuttle at kay mitochondrial NADH? Class, let's check the mechanism of the shuttles. So again, si MA shuttle, this is a mitochondrial inner membrane which is impermeable to NAD plus or NADH but is permeable to ions that attach to your malate. Let's see, diba? Si aspartate, as you can see here, is our initial molecule. So from your mitochondria, it will go outside to your cytosol via specific transporter, your glutamate aspartate transporter. Alright, so through transamination, i-convert natin yan sa oxaloacetate. And then si oxaloacetate will be converted to malate. Then here you can see, merong electron transfer. Sir, parang familiar yung second reaction. Is it just the reverse of the 8 reaction ng TCA cycle? Yes po. And this is very strategic because again, yung electrons from your NADH will be stored to your malate. Sir, bakit si malate ang gagamitin? Because malate has a specific transporter that can guide or that can help transfer your electrons from cytosol to mitochondria. Let's see. Si malate will enter mitochondria through your malate alpha-ketoglutarate transporter. And then, yung reverse reaction that happens in your cytosol will be occurring in your mitochondria. And as you can see here, we can successfully regenerate NADH via the conversion of malate to oxaloacetate. But class, please don't forget, kailangan i-revert back natin si oxaloacetate to aspartate for the next round of your malate aspartate shuttle. Diba? As you can see, we successfully transported cytosolic NADH to its mitochondrial NADH counterpart. So as you can see, na-retain ang original NADH electron carrier natin. Let's go to the glycerol 3-phosphate shuttle. For G3P, it requires 5 to transfer electron to your ubiquinone or abbreviated as Q for the electron transport chain. So as you can see here, ang ating main, ayan, ang key intermediate natin is si DHAP. So DHAP will be used as an electron carrier for your NADH to form glycerol 3-phosphate. And then si glycerol 3-phosphate will be processed by your G3PDH which is mitochondrial. And this will form FADH2. Sir, ba't nag-iba? Saan napunta sa NADH? Class, it is converted to FADH2 in order to regenerate DHAP for another cycle of your shuttle, your G3P shuttle. Kaya pala sir, 1.5 ATP lang ang conversion natin. Yes, because hindi na-retain si NADH. Because when it entered your mitochondria, it's in your FADH2 molecule. And now, these two molecules are now ready for oxidative phosphorylation. Alright, to summarize, these mitochondrial shuttles, we have this table available in your manual. So class again, location, MA shuttle, sa liver, kidney, and heart lang po sila localized. Si G3P shuttle, sa brain and skeletal muscle. In terms of the mechanism, ayan, again, si malic aspartate shuttle, this is a series of reaction, and in particular, a redox reaction exchange between cytosol and your mitochondrial matrix. So for your G3P shuttle, ito class, very straightforward. Direct electron transfer lang po siya from your DHAP to your FAD in order to generate FADH2 inside your mitochondria. O class, please familiarize our key intermediates. It's easy to remember for your malic aspartate shuttle. O, diba? Sa G3P shuttle, we have your glycerol 3 phosphate and we also have your DHAP. And then for our electron acceptor, very obvious na. For malic aspartate shuttle, NAD+. For G3P shuttle, si FAD po. And for membrane transport, of course, again, we have malic aspartate transporter for your malic aspartate shuttle. Kay G3P, sir, meron ba? None po. Again, kasi direct electron transfer ang mechanism ni G3P shuttle. And lastly, for cytosolic NADH, ang ATP yield natin for MA shuttle, 2.5. For G3P, approximately 1.5 ATP. But class, again, this is just an approximation because walang half ATP inside our cell. Okay, so that's it for our mitochondrial shuttle. So class, ngayon, lahat ng electron carrier natin is inside your mitochondria na. So ready na sila for our last process ng cellular respiration, which is your electron transport chain. Okay, so again, this is the process of your electron transport chain. First one, your reduced substrate donates electrons. So class, in particular, that's your fuel substrates, so your NADH and FADH2. So second one, electron carriers pump protons out as electrons flow to your final electron acceptor, oxygen. Okay, so sir, where is your electron transport chain located? So class, your ETC is located sa inner mitochondrial membrane. Going back, thirdly, the energy of the electron flow stored as electrochemical potential. So again, yung electron flow will generate an electrochemical potential. From the picture or from the diagram, inside or your mitochondrial matrix is negative, okay? While your intermembrane space is positive because there is your protons. Nandun yung proton gradient natin. And then lastly, your ATP synthase uses the proton gradient or your electrochemical potential in order to synthesize ATP. Okay, so class, that's the intro for our ETC. Again, to summarize, our reaction from your electrons and protons from NADH and FADH2, this will be transferred to our intermediate carriers. Again, class, kapag nagtatransfer tayo ng electrons, we also exchange or generate protons. That's why we were able to generate proton gradient because your intermediate carriers or your complexes pumped out your protons. And finally, ang electron acceptor natin at the last complex is your oxygen in order to produce our byproduct, water. And lastly, class, ayan. So here are the components of your electron transport chain. So again, we have 4 membrane-bound complexes as you can see here in our diagram. So we have complex 1 to 4. And also we have your unofficial complex 5, that is your ATP synthase. So some books kasi class, so some reference books, they treat ATP synthase as your complex 5. So aside from that, we also have your mobile electron carriers. So your coenzyme Q or your ubiquinone and your cytochrome C. So class, don't worry, we will discuss these components in detail here in our discussion of electron transport chain. O, diba? So here's a meme I found in Facebook. How are they working so hard without Spotify Premium? O, diba? Very busy ang complexes in your electron transport chain. Class, to formally introduce you the electron transport chain, let's watch this short video on ETC.
When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD+ and FAD are reduced to NADH plus H plus and FADH2. In the mitochondria, the electrons from NADH plus H plus are transferred to the electron carrier proteins, and the protons are transferred across the membrane. As the electrons move from cytochrome to cytochrome, down the electron transport chain, more protons are carried across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen, the terminal electron acceptor, and water is formed as the product. The transfer of protons generates a proton motive force across the membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
Alright, so class, again, very helpful yung animation in order for you to better understand our electron transport chain. Or you can see all of the complexes are embedded in your inner mitochondrial membrane. Again, we recall we have 4 complexes here and we also have your 2 mobile electron carriers, si coenzyme Q or ubiquinone and si cytochrome C. Alright, so class, si ATP synthesis here in your electron transport chain is driven again by your proton motive force. So first factor is the chemical potential or your change in pH. So as you can see again, si mitochondrial matrix, negative po siya. So si intermembrane space is positive, meaning acidic siya. Okay? So secondly, the electric potential, because of that chemical potential will be negative inside or in our mitochondrial matrix. Okay, class, here are the important information in the prosthetic groups present in our enzyme complex. So you can see here, for our first 3 complexes, they have their own prosthetic groups. For NADH dehydrogenase, meron siyang flavin mononucleotide. For our second complex, again, succinate dehydrogenase, wherein we encountered this enzyme on our TCA cycle. It's important that this enzyme has FAD or flavin adenine dinucleotide. Our third enzyme, ubiquinone, cytochrome C oxidoreductase, contain hemigroups or iron groups, and these 3 enzyme complexes need iron-sulfur centers in their structure for electron transfer. And lastly, si cytochrome oxidase, aside from the hemi or the iron groups, meron siyang copper as its prosthetic or cofactor. So again, class, we will discuss in detail each complex and mobile electron carriers on the succeeding slides. Okay, so here is our first complex of our electron transport chain. Si NADH dehydrogenase is very specific in processing your NADH electron transfer. Sir, nasan po siya sa visualization natin? So here is your complex 1. And here are the processes and proton pumping that occur in your complex 1. Okay, so class, for the process, we have your 2 electron transfer coming from your NADH electron carrier, and this is being passed by your complex 1 to the ubiquinone mobile electron carrier. So of course, ang magiging product will be your reduced ubiquinol. So sir, naka-ubiquinol na po ba yung 2 electrons? Yes po, and this will be carried to your complex 3. O class, please remember the number of protons pumped in your complex 1 upon electron transfer. And here, as you can see in our reaction mechanism, you need your cofactor, flavin, mononucleotide. Sir, bakit po sa complex 3 siya pupunta? Bakit hindi po sa complex 2? So again class, si complex 1, this is specific for your NADH dehydrogenase. Si complex 2 po, this is specific naman for your FADH2 electron transfer. O, diba recall, we reviewed or we discussed from your citric acid cycle na si succinate dehydrogenase, or we also call it your succinate CoQ oxidoreductase, as a very unique enzyme because it both participates in your TCA cycle and electron transport chain. Oh, let's see the process and the protons being pumped here. For the process again, this is the part, or this enzyme is a part of your TCA cycle which transfers 1 electron from your FADH2. Oh, it's very important that you remember from NADH 2 electrons. Kay FADH2, 1 electron transfer lang po ang nag-occur, thereby reducing your CoQ to your ubiquinol or CoQH2. Okay? So similar with your complex 1, we will deliver the electron, or we will transfer it from complex 2 to complex 3. As you can see class, ang ating coenzyme here is si FAD. Okay? And in terms of your protons pumped, none po. Again, there is no enough energy in order to create a proton pumping here in your complex 2. So I hope that's clear. Now, before we proceed with our next 2 complexes, let's first discuss your 2 mobile electron carriers. Okay class, when we say mobile, again, they are not embedded in your inner mitochondrial membrane. And this is very important for their roles. Again, kasi sila ang nagde-deliver or naglilink ng mga complexes here in your electron transport chain. Let's check their position in our chain. So here, si CoQ or si ubiquinone, it is located between your complex 1 and complex 3, as well as complex 2 and complex 3. So again, si CoQ can deliver 2 electrons. On the other hand, si cytochrome C, again, this links complex 3 to your complex 4. And later on, we will see the difference of these two with their respective capability on electron transfer. Class, here are the important definitions or properties of these 2 mobile electron carriers. Let's see. Si coenzyme Q, remember, this is mobile. It is non-protein and lipid soluble. It's very important na lipid soluble because again, it should be hydrophobic. Because again, it is on your inner mitochondrial membrane. And your mitochondrial membrane, similar with our bilayer, is relatively hydrophobic. So on the other hand, si cytochrome C, ayan, so it's a protein that contains an iron group, or we call it your heme group. So again class, when we say heme, collective structure po siya that includes porphyrin ring. So we have seen this, or we have seen the structure of your porphyrin in the citric acid cycle's anabolic role. So that's why it's very important as well na merong anabolic role sa citric acid cycle. So again, heme group includes porphyrin ring and your iron 3 plus center in order to stabilize the ring. Okay, so now I hope that's clear. So for our last 2 complexes, complex 3 and complex 4 complete the whole electron transport chain process. So, ayan, si cytochrome BC1 complex is also known your complex 3. On the other hand, we call cytochrome C oxidase si complex 4. So, class, these two complexes are somehow related, so they are in one family of your cytochromes. So here you can see there are similarities on their structure or reactions in particular. So as you can see, they both use your iron pairs, the iron 2 and iron 3 plus. And of course, ayan, they are on the last part of our chain, and here are the important information that you need to know. So starting, of course, with complex 3, so class, let's check from your CoQH2, we will oxidize that electron carrier in order to transfer your electron, and this will be passed via your complex 3 to your cytochrome C, which is another mobile electron carrier. Oh sir, may proton pumping ba here? Yes po. Okay, here we are able, or our complex 3 is able to pump 4 protons. Okay, and lastly, si complex 4, ayan. So this will just complete our whole electron transfer. Again, si complex 4 po is the location of our oxygen, the final electron acceptor ng ating electron transport chain. Let's check the process. From your cytochrome C, it will pass an electron to your complex 4. So similar mechanism with complex 3? Yes po. So this is a redox reaction between your iron 2 plus and iron 3 plus in order to give or transfer your electron to our final electron acceptor, oxygen. And sir, ang product po ba water? Yes. And here class, remember our proton pumping 2. So again, from complex 1 we have 4 protons, for complex 2 none, complex 3 we have 4, and lastly, complex 4 we have 2. So later on, we will have an accounting as well of the protons or the total protons pumped in our ETC. So here is the last complex. So our unofficial complex 5. That is your ATP synthase. Ayan, so class, I hope that you can see clearly the structure. Sir, do we need to memorize the components? No po. So I will just emphasize the important components of your ATP synthase. So class, here, you just need to know the 2 domains of your ATP synthase. So we have your F sub 1. So this is responsible in ATP synthesis. So class, this is an extramembranous domain. So you can see that it is not embedded. So again, extramembranous domain, meaning hindi po siya embedded sa inner mitochondrial membrane natin. So it is located on our mitochondrial matrix. Okay, connected by your B2 subunit. So class, again, you just need to remember si F sub 1, our ATP synthesis location. And secondly, si F sub 0, our proton translocation subunit. So here are the information that we need to know for our ATP synthase reaction. Ayan, so coupling reaction pala ang nag-occur with our ATP synthase enzyme. So here, in particular, this enzyme moves the protons that were pumped by your four complexes or three complexes rather out of the matrix. Okay, again, from your mitochondrial matrix to your intermembrane space. That's the direction of our proton pumping in our three complexes. O sir, ano po ulit yung domains? Ayan, we have 2. Si F sub 0, again, this is your membrane spanning domain. So meaning embedded siya in your inner mitochondrial membrane, which is responsible again for proton translocation. So class, as you can see here, there is an arrow. So here is the site of the entry of your protons. And this is like a rotor. It will rotate and this will power your F sub 1 domain for ATP synthesis. Sorry. So for ATP synthesis, again, using your coenzyme, ADP, and another cofactor in organic phosphate. So sir, ano po ang mechanism here in ATP synthesis? So class, similar with the process, coupling din po with your chemiosmosis and oxidative phosphorylation. So for our next slide, we will formally define these two mechanisms. Again, this is your whole electron transport chain. And as you can see, somehow, your ATP synthase or complex 5 is far in terms of its location with our four complexes. So class, you can clearly see here in our visualization, the proton pumping, the charges of your mitochondrial matrix, and your intermembrane space. Also, what are those transport proteins that are also embedded here in our inner mitochondrial membrane? Class, ang transport proteins rather are very important to deliver the coenzymes that are needed by our ATP synthase. Okay, so we have several active transport mechanism here that occurs in our complex 5 or ATP synthase. So here we have two active transporter. Ayan, so as you can see, for our left visualization, si chemiosmosis po. So again, chemiosmosis is just the movement of your ions, particularly your protons, across a semi-permeable membrane. So in this case, your inner mitochondrial membrane. So here, let's briefly discuss the active transport mechanism. First one, we have your adenine nucleotide translocase. So sir, specific po ba yung transport protein na yan? No po. So as you can see here, we label it as antiporter because it can transport your proteins in both directions. Okay? So here we can see going into our mitochondrial matrix is your ADP, while going out is your ATP generated by your ATP synthase. So it's important kasi that we have a specific transport protein for this energy currency of the cell because again, it's important that we localize or mobilize efficiently our ATP for cellular needs. Alright, so secondly, ayan, so sir, what is that next translocase? So this is your phosphate translocase. Again, this is important for the reaction of your ATP synthase. Okay, so at class, it's important kasi that we maintain the acidic environment here in your intermembrane space and the negative charge here in our mitochondrial matrix. Alright, so now here are the formal definition of the two processes here in our ATP synthase. So again, I already discussed chemiosmosis. So just the movement of ions across a semi-permeable membrane. So again, when we say down, their electrochemical gradient, sir, ano nga po ang direction from high to low concentration? Alright, so for oxidative phosphorylation, comparing it with SLP again, to substrate level phosphorylation is the direct transfer of your phosphate group from your original substrate to our final product or final substrate. Here, OP is the process in which ATP is formed due to the electron transport or electron transfer process. Okay, so again, coupled siya with your chemiosmosis process here in ATP synthase. Class, please again remember our four complexes plus ATP synthase here in our electron transport chain. The processes, the proton pumping, the different active transport occurring, and the processes here in our ATP synthase, namely chemiosmosis and oxidative phosphorylation. Sir, summarize na natin si electron transport chain. Okay, so again class, the location of our electron transport chain, particularly the complexes, complex 1 to 4, and your ATP synthase, they are embedded in your inner mitochondrial membrane. Let's summarize the different enzymes or complexes along with the process and their proton pumping. First one, for our complex 1, again, electron transferred, we have 2, and flow ng electrons natin from NADH to your ubiquinone with the help of the cofactor flavin mononucleotide. And remember, here we pump 4 protons out of our mitochondrial matrix to your intermembrane space. Class, moving forward with complex 2, our succinate dehydrogenase or your succinate CoQ oxidoreductase. Ayan. So here, again, only 1 electron is transferred from your FADH2 to your similar mobile electron carrier ubiquinone. Sir, may proton pumping, again class, none because of the lack of enough energy by this complex. Thirdly, we have your cytochrome BC1 complex. Similar with our first complex, electron transferred here, dalawa, and your protons pumped 4. But here, our flow of electrons from our mobile electron carrier, ubiquinol, the reduced form of your CoQ H2 to your cytochrome C. Okay, again, the cytochrome C is another mobile electron carrier. And lastly, we have cytochrome C oxidase, similar with our complex 2, only 1 electron is transferred from your cytochrome C to our final electron acceptor, oxygen. So here class, of course, we need 2 electrons for your oxygen. That's why twice po nagyayari ang electron transfer here from cytochrome C to your oxygen. That's why protons pump, dalawa din po. Alright, for ATP synthase, again, our processes include chemiosmosis and oxidative phosphorylation. Okay, class, don't forget our mobile electron carriers. Diba? As I have said earlier, si CoQ and cytochrome C, they are different in their electron transfer capability. Si CoQ or si ubiquinone can carry 2 electrons at a time. Si cytochrome C, only 1 electron at a time. So please remember this very essential information. Ayan, so similar with our discussions with the 3 processes of your cellular respiration, of course, we have a regulation discussion here in electron transport chain. O sir, parang may new character. Yes po. So in electron transport chain, we have 2 different regulators, si inhibitors and si uncouplers. First, si inhibitors, these block complexes through binding with specific electron carriers. Sir, ang target nyo ba is si complex 1 until ATP synthase? Yes po. Alright, so here are the specific inhibitors of our 4 complexes plus ATP synthase. Class, please memorize. So I require you to memorize the inhibitors because these appears in your board exam. Okay? So very few lang naman ang common inhibitors ng ating complexes here in ATC. So first one, for complex 1, si metformin. So a drug for your diabetic patients, diba? So remember, of course, this has something to do with our glucose mobilization. Next, we have rotenone and pyridine A. For complex 2, we only have one, simalonate. For complex 3, ayan. So class, remember the 2. This always appear as well in your board exam. So if I remember correctly, 2022 boards, nag-appear ang question na to. So we never know if they will reuse this specific question. So complex 3, we have dimethylcopperol and antimycin A. Complex 4, ayan. So familiar with this 2 first 2. So we have cyanide and carbon monoxide, which are both irreversible inhibitors as we have discussed in our discussion on enzyme, diba? So in your different types of inhibition. So thirdly, we have your dihydrogen sulfide. And lastly, for ATP synthase, si oligomyosin. So class, si oligomyosin, it binds to our F sub 0 domain ng ATP synthase. So siya ano po ang consequence? Or recall class, si F sub 0 is for proton translocation. Thus, ayan, kino-close niya ang proton channels natin. That's why nagkakaroon ng build-up ng protons in our intermembrane space. Alright, so now, sir, ano po ang uncouplers? So si uncouplers, it disrupts membrane permeability. So usually, si inner mitochondrial membrane. Okay, so this is done via the dissipation of your proton gradient. So class, ang nature or ang structure po ng uncouplers are very similar with our ATP synthase. They also have their different proton translocation subunit, and that's why ang target, okay, ang target po ng uncouplers ay ATP synthase only. So sir, meaning yung proton gradient translocation lang po ba ang affected here? Yes. So again, si uncouplers somehow, it does not directly or it does not affect in general our electron transfer. So later on, you will see pa nga, like, na they prefer an increase in electron transfer of our complexes. Class, let's see the two different types of uncouplers. First one with chemical, ayan, very known si dinitrophenol to be an uncoupler of our ATP synthase. Okay? So class, remember dinitrophenol, dicumarol, and CCCP, or your carbonyl cyanide and chlorophenyl hydrazone. No need to memorize the structure. And secondly, we also have your biological uncouplers, or we call it in our biological systems uncoupling proteins. Class, ang biological uncouplers, this is not actually a disadvantage for us, particularly on hibernating animals, because this serves as their insulation in winter season. So for instance, we have thermogenin. Class, this uncoupler is very known to appear always in the board exam. So in the 2023 boards during our time, nag-appear din po siya. Similar with 2022. So again, si thermogenin is found biologically in our fat tissue or brown adipose tissue. So of course, this is an endogenous protein. So meaning we synthesize this actually. So again, this acts as an coupler which uncouples your ATP synthesis from electron transport chain. So CUCP thermogenin has its own proton translocation subunit, and of course this is found in tissues of newborn animals and hibernating animals. Okay, so to summarize again our discussion on electron transport chain, so remember our tabulated inhibitors and uncouplers. Class, here as I think, I hope that you can see this clearly. This is your uncoupling protein such as thermogenin. As you can see, it provides an alternative proton translocation, thus it uncouples your proton gradient with ATP synthase. That's why ang result, dissipation ng proton gradient natin. So this is very inefficient in terms of energy reduction. Okay, so to end our discussion on electron transport chain, ayan, so we summarize the effects of inhibitors and uncouplers in the processes occurring in our electron transport chain. So let's discuss this in detail. First one, the electron transport. So class, for inhibitors, remember, their targets are your complexes. And complexes, remember, as we have discussed, they are responsible on or in electron transfer from complex 1 until it reaches complex 4 for our final electron transfer to oxygen. So sir, meaning may inhibit po ng inhibitors ang electron transport? Yes po. Okay, so I hope that's clear. For our uncouplers, so the opposite is true. So remember class, ang uncouplers, they provide an alternative proton translocation subunit, thus mas preferred nila ang higher proton gradient. O remember, sino ba ang nagkakreate ng proton gradient? Yung proton pumping by your complexes. Thus, mas preferred ng uncouplers to increase your electron transport. Mas gusto pa nga nila na mas increase ang activity ng complexes. So again here, ang relationship, electron transport will increase in the presence of your uncouplers, because in turn, ang result ng increased electron transfer is increase as well of your pumping ng proton gradient, and here, more available protons will be an advantage of your uncouplers. Okay, for ATP synthesis, ayan, both inhibitors and uncouplers will inhibit or will stop ATP synthesis, because for inhibitors, wala po tayong electron transfer occurring. For uncouplers, again, it provides an alternative inefficient proton translocation process. Okay, so again, inactivated po in this case or in the presence of uncouplers, si ATP synthase. And thirdly, we have oxygen consumption. Let's check sa inhibitors again, because inhibited po ang ating 4 complexes. Of course, hindi makakarating sa oxygen, our final electron acceptor, ang electrons natin. Thus, there will be a decrease in oxygen consumption. Okay, so I hope that's clear, class. So very straightforward ang relationship ng inhibitors with oxygen consumption. For uncouplers, ang oxygen consumption will increase. Okay, so remember our discussion on electron transport, si oxygen consumption ay mag-i-increase due to the fact na of course, nag-i-increase din po kasi ang electron transfer natin in order to increase the proton gradient ng ating intermembrane space. Alright, so here, again, for uncouplers, it prefers an increase in oxygen consumption. And lastly, for our OP and ATP synthesis, ayan, against inhibitor, energy-rich, ATP and activator, energy-deficient, ADP and your inorganic phosphate. Alright, so to summarize our discussion, or to end our discussion, we have a segment here in our electron transport chain. Alright, so electron transport chain is actually one of the sources of our aging-related damage in the body. So class, again, this is a natural process. You can slow it down, but you cannot stop it. Alright, so why, sir? What's the reason? So again, class, as electrons travel down your electron transport chain, some of the electrons escapes and react with oxygen, creating ROS, or reactive oxygen species, as byproduct. So for instance, we have your superoxide. So here we will see in our discussion on pentose phosphate pathway how to neutralize or how to treat the production of ROS. Okay, so thus over time, this side reaction causes oxidative stress in our cells that damages nucleic acids and other biomolecules that contributes to our normal process of aging, neurodegeneration, and sometimes even cancer. O, diba? So again, this is one of the natural processes or natural degradation process that occur inside our cells. And of course, it has different effects. So next, ayan, bioluminescent organisms glow by hijacking ETC or electron transport chain like chemistry. O, di ba? Amazing. So here are some examples of your bioluminescent organisms, include your fungi and your algae. Okay, so class here, your bioluminescent organisms use reactions that are similar to your electron transfer reactions in electron transport chain all the way to your oxygen. So here, instead of generating ATP, they use the energy in order to emit light. And here, ano po ang main character enzyme natin? Si luciferase. O, di ba? For instance, sa fireflies or our deep sea squid, they use luciferase to mimic your electron transport chain energy transfer here in order to power their glow in the dark display. So here is the specific reaction. No need to remember, no need to memorize. Okay? So class, this is the end of our discussion on electron transport chain. O sir, pwede na po ba tayong mag-ATP accounting since we are already done with all four processes of cellular respiration? Okay po, so here is the ATP yield calculation. Okay, so here are some important reminders for you to know in our calculation. Now remember, since in our TCA cycle, our original substrate is acetyl-CoA. And acetyl-CoA is originates from your pyruvate. And recall, one glucose produces two molecules of your pyruvate. Hence, need nating i-multiply by 2 ang TCA cycle products. Alright? So here in our proton transfer along the complexes in ETC, so class remember, di ba? For our NADH, ang nagpa-participate, complex 1, 3, and 4. Okay? That's why we have a total of 10 protons pumped for NADH. So 4 protons from your complex 1, 4 protons from complex 3, and 2 protons from complex 4. On the other hand, 6 protons lang kay FADH2 due to the reason na there are no protons pumped by complex 2. So again, ang 6 protons are just coming from your complex 3, 4 protons, and complex 4, 2 protons. So for ATP synthesis, ayan. So here is the rough conversion of your ATP with your protons pumped. So here, 1 ATP is equal to approximately 4 protons pumped in our ATP synthase. So thus, taking into account the total number of protons for each electron carrier NADH and FADH2, we have 10 divided by 4 for NADH equals 2.5 ATP and 6 divided by 4 is equal to approximately 1.5 ATP. Kaya pala sir, in our glycerol 3-phosphate shuttle, si FADH2 ang conversion ay 1.5 ATP. Yes po. Okay, so to summarize, ayan, so again, here is the total ATP yield for our two different shuttles. Let's check. For glycolysis again, net yield natin 2 ATP. That's why for both shuttles, we only have 2 ATP. Okay, so again, sa glycolysis, we produce 2 NADH, which are cytosolic. O sir, yan ba yung NADH na need ng shuttles? Yes po. Class, remember in our discussion in mitochondrial shuttles, ang rough conversion po for our malic aspartate shuttle, again, 1 cytosolic NADH will be converted roughly to 2.5 ATP. That's why 2 times 2.5 is equal to 5. On the other hand, the G3P shuttle again, 1 cytosolic NADH will be converted to 1.5 ATP because again, upon transfer of your NADH inside the mitochondria, we form FADH2. That's why 2 times 1.5 is equal to 3. Okay, so for our second reaction, your pyruvate conversion via oxidative decarboxylation to your acetyl-CoA, we generated 2 mitochondrial NADH. And these NADHs are readily oxidized. That's why 2 times 2.5 ang conversion natin equal both to 5 ATP. Alright, so for the citric acid cycle, for MA and G3P shuttle, same lang po ang yield because again, your shuttles here do not affect your TCA cycle energy products. So let's see. Here is the list of our energy products from our TCA cycle. And remember, we need to multiply it by 2 to get the net yield of ATP per mole glucose. Alright, again, class, emphasis on per mole glucose po kasi ang basis natin. Wala namang 1/2 glucose inside our cell. Let's check. 3 NADH for 1 round of citric acid cycle times 2 again because we generated 2 pyruvate that will be converted to 2 acetyl-CoA. So again, we will get 6 NADH times 2.5. Ayan, we have 15 for your MA and G3P shuttle, the net yield of ATP. Secondly, we have 1 FADH2 again multiplied by 2 times, ayan ang conversion class, 1.5 ATP, so 2 times 1.5, both of them, 3 net ATP yield. Ayan, so class, here we can see that 1 GTP times 2 is equal to 2, of course, both for your MA shuttle and GTP shuttle. Class, let's sum up all our ATP yield. So 2 plus 5, 7, plus 5, 12, plus 15, 27, plus 3, 30, plus 2, 32. Okay, so that's for your MA shuttle. May additional tayong 2 ATP because sa GTP, we only obtained 30. So class, again, ang ating ATP yield is dependent on the localization of your shuttles inside the body. So class, ang ating range of ATP yield is around 30 to 32 ATP. Class, remember na ha, para hindi na kayo magko-compute sa mismong board exam for your glucose. Kapag maliit aspartate shuttle, 32. For G3P shuttle, 30. Alright, so here is another way of calculating our net ATP yield. Plus, please remember again, 1 glucose molecule yields 30 to 32 ATP. Sir, kapag ang question po ba is to glucose, we just need to multiply it by 2? Yes po. And please remember ha, meron kasing specific shuttles. Again, 32 for malic aspartate and 30 for our G3P shuttle. Alright, so class, we are now done with our cellular respiration discussion. Congrats for finishing this video. As a reward, ayan, we have a quick quest to test your knowledge on our Crash Course 6, particularly here in our electron transport chain and TCA cycle. For our first set of concepts, ubiquinone, ATP synthase, cytochrome C oxidase, and PFK1. Ay, sir, ang obvious naman, dahil na tayo sa glycolysis wala na dapat si PFK1. Okay? So ubiquinone, ATP synthase, and cytochrome C oxidase all participate in our electron transport chain. Ayan, and last, we also have G3P shuttle, malic aspartate shuttle, pyruvate dehydrogenase, and NADH. Class, recall our discussion on mitochondrial shuttle, which is not related. Add one out, a forest answer, pyruvate BH. Alright, so this is under our discussion of fates of pyruvate. Diba, see, pyruvate BH is the enzyme responsible in oxidative decarboxylation of pyruvate to yield acetyl-CoA. Alright, so these are my final Lars words for our cellular respiration discussion. Class, similar with biomolecules, please check your biochem notes at the end of the discussion on amino acid metabolism. We have a summary of your different metabolic pathways starting with cellular respiration. Okay, again for glycolysis, sir, ang dali ng glycolysis pala, that's just conversion of 1 glucose to 2 moles of pyruvate. And remember, catabolic po against glycolysis because again, we were able to generate energy in the form of ATP. And location, cytosol. Ayan. Class, remember the regulated enzymes and the relevant energy products. Pyruvates, 2 ATP and 2 NADH per 1 mole glucose. Second process, fates of pyruvate, we have 3 different processes here. For fermentation, we have 2 types, alcohol and lactic acid, both of them catabolic, both of them occur in your cytosol. Class, again, similar with glycolysis, you need to remember the key enzymes here as well as the relevant products. So I will not discuss this in detail because we already discussed this in detail in our discussion earlier. Diba? So thirdly, we have aerobic conversion of set of pyruvate, to your acetyl-CoA through oxidative decarboxylation. Remember, class, catabolic din yan because we are able to generate NADH. And sir, cytosol pa rin po ba? Again, class, no. It is inside your mitochondrial matrix. And here, class, again, very unique ang enzyme because that is the committed step of your TCA cycle yielding acetyl-CoA, NADH, and your byproduct, carbon dioxide. Malapit na tayo matapos. So thirdly, with our citric acid cycle, we oxidize here your acetyl-CoA to generate a large amount of energy. Again, class, TCA cycle with your electron transport chain, they are the central hub of energy yielding pathways here in our metabolic pathways discussion. Remember, we discussed the metabolic and anabolic role of citric acid cycle because again, this cycle is amphibolic occurring in your mitochondria. Similar with glycolysis, remember our regulated enzymes. Diba? We discussed the inhibitors, the activators of these enzymes, as well as their deficiencies if applicable. And lastly, as we have computed in our net ATP yield calculation, remember our energy products, ATP, 3 NADHs, and 1 FADH2 with generation of 2 carbon dioxide from your 2 oxidative decarboxylation reaction of first one, isocitric dehydrogenase, and second one, alpha-ketoglutarate dehydrogenase. Alright, so class, this is the last process under our cellular respiration. So this is your electron transport chain. Hi, salamat sir, makakahinga na rin kami. Class, very fresh ang discussion natin here. Again, process is pretty straightforward. Electron transfer for energy generation and for water production. Remember, catabolic po siya because we produce a large amount of energy and location inside your inner mitochondrial membrane. We have regulated enzymes here as well. Si complex 4 and complex 5 are somehow that tightly regulated because as we have seen in our regulation discussion, they have a lot of inhibitors. Alright, so I hope that's clear. And lastly, we have your relevant products. So again, we are in respect with your 1 mole glucose. Here, 1 mole glucose generates 30 to 32 ATP with water as your byproduct. Alright, so class, here's a meme that I have seen. So this is in terms of energy production ng processes in your cellular respiration. Diba, relatively, Krebs cycle, electron transport chain have generated or they were able to generate large amounts of energy in the form of ATP. On the other hand, in glycolysis, we have seen it can only generate at most 2 ATP per glucose molecule. To summarize and to conclude our discussion in this video, here is our review checklist. So class, again, this is under your respiration process in the cell. So we were able to discuss and identify each pathway and its metabolic type. We also tackled the cellular locations of the 4 major processes, and of course, hindi nawawala ang discussion on regulated enzymes and their inhibitors and activators. We also discussed the deficiencies of these enzymes, and fourth, we also associate steps with your major energy products. Alright, so lastly, we also compare the outcomes of each metabolic pathway, particularly sa fates of pyruvate in terms of oxygen availability. Class, here's the summary of the mnemonics that I have shared in our discussion. So again, feel free to use the mnemonics. We will compile all the mnemonics that will be helpful in your whole review duration. So with that, ready na ba mag-answer ng PT number 6? So class, again, please practice closed manual answering so you can review first our discussion on citric acid cycle, mitochondrial shuttles, and electron transport chain, as well as ATP accounting before you answer PT6 10 items for 15 minutes. Alright, so we are now done with our cellular respiration. For our next video, diba may teaser, for CC7, we are going to cover your carbohydrate metabolism. Alright class, study well. Bye!